Good morning, ladies and gentlemen. Now we are going to start our scientific program of IBBC 2023. I feel privileged to extend my warm welcome to all of you for our first International Brain and Biomedicine Conference, IBBC 2023. Let us now proceed our today's event with the plenary session. It's going to be very informative. I sincerely hope that all of you will learn from this session. To kickstart the plenary session, now I'm honored to invite our first keynote speaker, Professor Dr. Mohsin Kanak, Vice Chancellor, Askıdar University, Turkey, and he will talk about some features of hesperidin. Please, Professor Dr. Mohsin Kanak.
Hello, everyone. First of all, I'd like to thank to the organizing committee to invite me to give a talk. It's going to be around 20 minutes. And I'm going to talk about the hesperidin and some important features of the hesperidin. I'm Professor Mohsin Kanak from Üsküdar University, which is the Turkey's brain base. Actually, our university is one of the first thematic university studying on the mainly behavioral and health sciences. Therefore, this is our motto, and this is the actually brain base of Turkey, because Üsküdar University has attended to Mr. Obama's Brain Initiative project, and we are coordinator of Balkan and Middle East countries. Maybe next time we can organize these meetings, as joint conference. This is my topic is on some features of the hesperidin. And the hesperidin, as everybody knows, is a flavonol glycoside found in the citrus fruit. It is a glycon form is called hesperetin. Actually, as you see here, and this is the hesperetin. And when it is, added to sugar, then it's becoming hesperidin. Hesperidin was first isolated in 1828 by French chemist Lebreton from the white inner layer of the citrus peels. Actually, it could be mesocarp or albedo, and it is believed to play a role in the plant defense. Scavenging the free radicals reveals its antioxidant property. Hesperidin is known for its antioxidant, anti-inflammatory, and neuroprotective effects, and provides modulation drug metabolizing enzymes without the toxicity. The biosynthesis of hesperidin is starting from L-phenylalanine in 9 steps, as you see here, and this is the biosynthesis in the citrus fruit. Actually, I'm going to present some of our research, and at the end, finally, I'm going to share some research done by others and published in a very, highly impacted journals. This is, the mainly the explanation of our projects and our studies. And for these studies, actually the ethical, because we did the experiments on the mainly on the rats, and we got the ethical approvals for each study obtained from Üsküdar University Experimental Animal Ethical Committee before beginning the experiments. One of the first study, actually, it is in silico study, and it is published in the journal Biological Physics, and the title was the radiation shielding parameters of some antioxidants using Monte Carlo method. In this study, we aim to investigate the attenuation behaviors of antioxidant samples such as delphinidine chloride, ferulic acid, asparagine, epicatechin, gallate, p-coumaric acid, quercetin, narireutin, and camphorol against different photon energies of some radioisotopes in novel Monte Carlo simulation setup using MCNPX. Actually, it is the acronym of Monte Carlo and particle extended code. The results show that on average, the highest and lowest radiation mass attenuation coefficients were observed on hesperidine and ferulic acid, respectively, when they compared to the others. It can also be concluded that the obtained results from this study are very useful for radiology and radiotherapy applications where antioxidants are frequently used. And when you can see the graphs with the ferulic acid and the hesperidine, and they are almost more or less the same result, and they are decreasing the attenuation, the radiation effect on the tissues. Because of its frequent use of different biological and pharmacological systems, especially the therapeutic application due to its radiation reducing properties, this study can be very beneficial to the scientific community. For future studies on related subjects, in summary, the investigated physical principles of radiation protection and the guidelines of ALARA, combined with the biological protection provided by multiple antioxidants, may further decrease the health risk of low doses of radiation, no matter how small that risk might be. Another study actually we did in our lab, and we published in the Fresenius Environmental Bulletin, and the title of the paper was Antioxidant Effect of Hesperidine on Cataract Rat Pups. The aim of this study was to investigate the effects of hesperidine on oxidative damage to lens tissue in a cataract model using sodium selenite. And as you see, the results actually in the graph first, and you can have a look at the GSH and MDA and total antioxidant status. So this is showing the control group, in the first block, then sodium selenate and the hesperidine, and the hesperidine plus sodium selenate. And as you see here, and there was really a big and significant importance in all tests we examined. And this is the picture of the eyes of the rats, and this is the control group, and without giving anything. And this group has given the hesperidine, and this group is only sodium selenate. and this group is sodium selenite plus HES. So as you see here, the difference in the eyes, and the hesperidin is really reducing the formation of the cataract on the eyes of the rats. In the another study, actually, we made an epilepsy model stimulated with pentyl and tetrazole, and we looked at the effect of the hesperidin and valproic acid combination, and we did have a look at the oxidant and antioxidant defense mechanism in the rats as well. And in this study, we aim to investigate the effects of combined use of valproic acid and anti-epileptic drug. It is the valproic acid with hesperidin on antioxidant defense system, both in brain and liver tissue specimen. For these purposes, a total of 48 3-month-old and 200 and 250 gram male Lister albino rats were divided into 6 groups, and in the 6th group, the group 1 was a control group, and in the group 2, pentyl netrazole, it's actually 60 milligram per kilogram given intraperitoneal. In group 3, hesperidin 100 milligram per kilogram is given orally by gavage, and in the group 4, pentyl netrazole and hesperidin, and in the group 5, pentyl netrazole plus valproic acid. Actually, they're the 400 milligram per kilogram interperitoneally. And in the group 6, pentyl netrazole plus valproic acid plus hesperidin. The results were subjected to statistical analysis by employing SPSS software, and the P equal or less than 0.05 accepted as significant value, and chemical agents were given to all groups outside the control group at 1st, 7th, 14th, and 28th days. Glutathione, malondialdehyde levels, and the superoxide dismutase activity were determined in the brain, and the liver tissue homogenates obtained at the end of the experiment. Our results demonstrated that hesperidin may increase antioxidant activity when used in combination with anti-epileptic drugs, and may reduce the potential side effect of the valproic acid alone. These figures are showing the results of our studies, and as you see here, in all of them, this is showing the glutathione, reduced glutathione levels, and this is the actually obtained from the brain, and this is this result obtained from the liver, and this is the MDA results from the brain, and MDA results from the liver tissue of the rats examined. And this is SOD levels from the brain, and this is the SOD results from the liver, and this is the just seizure level in the all the groups, and it was showing the 1st, 7th, 14th, and 28th days. Our results actually demonstrated that hesperidin may increase antioxidant activity when used in combination with anti-epileptic drugs and may reduce the potential side effects of valproic acid alone. This study actually has been accepted for the publication in the Sigma Journal of Engineering and Natural Sciences. It is in the Emergency Citation Index journal, and the title was The Effect of Hesperidin on Neuron-Specific Enolase and Oxidative Damage in the Preeclampsia Model. The objective of the study was to inspect the relation between preeclampsia and autism spectrum disorders and to examine the effects of hesperidin on the neuron-specific enolase levels and oxidative stress markers during preeclampsia and or risk of the autism spectrum disorder. Actually, this is unpublished data, and we studied on the rats again, and the study titled as has improves oxidative damage in an experimentally induced non-alcoholic fatty liver. And as you see here, this is the normal liver, and this is the fatty liver, and after that, the fatty liver plus inflammation had, seen in the liver, then it is going to cirrhosis. In this study, we established NFLD model by using high-fat, high-sugar diet and investigated the concentration of the oxidative stress markers on the liver of male Western albino rats. For these, rats were divided into 4 groups and 8 rats in each. And the first group was the control. Second was the control plus hesperidin. Actually, it is 150 milligram per kilogram per day. And NAFL, actually, it's high-fat, high-sugar diet, and NAFL plus hesperidin, high-fat, high-sugar diet plus 150 milligram kilogram per day. At the end of the experimental period, serum levels of fasting glucose, triglyceride, cholesterol, liver enzymes, and hepatic glutathione and malondialdehyde levels were measured. And later on, we have seen this graph. Actually, this is the showing glucose level, and this is triglyceride level. This is cholesterol level, and this is HDL cholesterol, LDL cholesterol, AST, and ALT. Actually, you can see the color of each group, and the first one and the blue one is showing the control group, and the yellow one is showing the cholesterol and the HES group, and the red one is showing NAFL, and the green one is NAFL plus HES group. Actually, you can see some asterisks here, some stars, and these are showing if it is only one star is there, and it is P value is lower than 0.05 as compared to the control, and cholesterol plus HES group. In cholesterol and HES group, actually, the P level is less than 0.001 as the compared C plus HES group, and you can see the effect of the hesperidin on the reducing of the the fat level in the liver. And this is the MDA and the control and CHS group. And as you see here, the GSH, and this is the showing the MDA group, as you see here. And actually, again, the left one is the control group, and the brown one in the second one is the control has, and this is the NAFL, and the dark blue one is NAFL and the S group. Actually, these are some articles, review and study articles published in the very famous journals, and you know, this is not these results are not our studies, but these are related with the protective effect of the hesperidin on the nervous system and the brain tissues, and the, you know, the benefits of hesperidin in central nervous system disorder is a review, and this is again hesperidin as a neuroprotective agent, a review of animal and clinical evidences, and it is a review again, and this is the published as article in the Nutrients Journal. Hesperidin improves memory function by enhancing neurogenesis in a mouse model of Alzheimer's disease. Actually, I'd like to thank all of you, and sorry I couldn't get any question from the presentation due to my you know, intensive work in Turkey, and I couldn't come to Pakistan. One day, hoping that and wishing that to come to Pakistan and visit my dear colleagues like Ghulam Hussein and the other colleagues. And again, I am suggesting the organizing committee to organize these meetings with Pakistani and the Turkish side all together. Thank you very much for your patience.
Thank you, Professor Dr. Mohsin Kanak, for such an amazing talk. It's a time now, ladies and gentlemen, for me to invite our second keynote speaker, Professor Dr. Hing Sohan, Chairperson, Department of Biomedical Sciences, Kyungpook National University, South Korea, and he will talk about NGS study in neurological diseases. Please, Professor Dr. Hing Sohan.
Hello everybody, I'm Hyung Soo-han from Korea. I'll give a talk about the NGS study in neurological diseases. The contents will be as follows. First, introduction to medical NGS. The history of sequencing is very long. From the beginning, when Crick and Watson discovered the DNA structure, sequencing of the DNA was tried. As you can see, many genome assembly was founded. Sequencing tools also was developed. Sanger sequencing, which is still used frequently, is kind of a first generation. And NGS, next generation sequencing, and now third generation of sequencing method is used. NGS is applied in several fields. First, DNA sequencing using whole genome sequencing. Whole genome sequencing is a comprehensive method to analyze an entire genome. It can detect single nucleotide variants, insertions, deletions, copy number changes, and large structural variants. So it is ideal for discovery of variants and novel genome assembly. As you can see, human genome consists of gene and intergenic regions. Whole genome sequencing covers all of these genome area. When we do whole genome sequencing, making reference genome sequence is the first thing we have to do. Based on the reference genome, we can identify individual genome sequences. Usually, reference genome sequence is the work achieved by government or big academic consortium, and researchers use the reference genome for their individual research. Human Genome Project is the first international collaboratory research program, which designed to complete all the genes of human. Based on these accomplishments, we can initiate many of the researches on human genome. Reference genome is DNA blueprints used as a standard for comparison in basic research and clinical settings. The current version of reference genome is called Genome Reference Consortium Human Build 38. If you visit Genome Reference Consortium site, you can see or download the reference genome data. Second part of the DNA sequencing is whole exome sequencing. Exome is the genome that takes all the exons together. It consists of 1 or 2% of the whole genome. So if you are interested in the genes which cause proteins, it's better to perform whole genome sequencing. Whole genome sequencing process is similar to whole exome sequencing. The only difference is you have to capture whole exome using capture process. The other processes are similar to whole genome sequencing. Targeted sequencing is next generation sequencing, which elucidate some defined small number of genes. Due to high depth of targeted sequencing, it is commonly used in the clinic, but it has some limitation. When we do targeted sequencing, we usually make some multi-gene panels. The targeted sequencing has higher depth and wider coverage, so it is very suitable for clinical application, especially when you have good biomarker panels compared to whole genome or whole exome sequencing. Gene panel usually includes 10 to 1,000 biomarkers. This figure shows the brief comparison of whole genome sequencing and whole exome sequencing and target sequencing. RNA sequencing is a method which examines the quantity and sequences of RNA in a sample using NGS method. It analyzes the transcriptome, so it shows the information about the genes. RNA-seq is one of the most commonly used NGS method. As you can see from the figures, RNA-seq experimental steps includes sample collection, isolation of RNA, and making cDNA, and the following steps are similar to a DNA sequencing method. These days, single-cell RNA sequencing is commonly used. You can take tissue and isolate and sequence individual cells. Then you will have RNA expression analysis of single cells. Spatial RNA-seq is another method derived from the single-cell RNA-seq, but it provides information on location. Few years ago, I started the NGS study. Whenever you are a beginner, you have to know some concept of NGS before starting your study. First, Sanger sequencing versus NGS. In principle, the concept of Sanger or NGS technologies are similar. This figure shows the steps of Sanger versus next generation sequencing. The most critical difference between Sanger sequencing and NGS is sequencing volume. Because Sanger sequencing can sequence one DNA fragment at a time, while NGS deals thousands or millions of fragments simultaneously. Since Sanger sequencing has the ability for longness and gold standards of accuracy, it is perfect for sequencing single genes or sequencing small amplicon targets at the lowest cost, or sequencing up to small samples at a time, like microbial identification or fragment analysis or the genotyping. And it is very important to be used for the confirmation of NGS result. I also use Sanger sequencing to confirm novel finding. NGS offers fast turnaround time, and it takes very short time to complete the study. So NGS is perfect for interrogating larger number of genes at a time and finding novel variants, or you can sequence samples with a low input amount of starting material. Sequencing microbial genomes for pathogen subtyping is also can be used using the NGS analysis. Variant analysis is one of the most important and most commonly used study method in clinical medicine. We can use three different formats: quantitative PCR, or Sanger sequencing, or NGS. Usually it is targeted sequencing. Microarray is another option we can use to study variant analysis, but now NGS replaces the position of microarray. These three methods has its own benefits and limitations. For qPCR, benefit includes high sensitivity, quick and simple workflow, and capital equipments because PCR machine is already found in most labs. But it has limitations, including only examine a small set of variants, virtually no discovery power, low variant resolution, low scalability. If you use targeted NGS, It has benefits including expanded discovery power, higher analytical sensitivity, greater resolution, and etc. So you can choose one of these methods for your study by comparing the benefits and limitations. Sanger sequencing and qPCR is good choice if you need to interrogate small region of the DNA on a limited number of samples. Otherwise, targeted NGS is shoot for your needs because it allows you to screen more samples and detect multiple variant types across targeted area. We can compare three DNA sequencing methods: whole genome sequencing, whole exome sequencing, and targeted sequencing. By comparing the benefits or limitations of this method, we can apply one of these methods for your study. Usually, targeted panel sequencing has higher accuracy, while whole genome sequencing has lower accuracy. Whole genome sequencing sequences whole genome, so it is very good for discovery purposes, but the sequencing depth is lower and it covers everything, so it can identify all kinds of variants. Targeted sequencing just targets small specific regions, and the sequencing depth is very high, like 500 or 1000. It also can identify all kinds of variants, and as long as you know the targets well, it is the most cost-effective method. Since three different RNA sequencing methods has different limitations and benefits, depending on the gross features or location information or resolution, you can choose one of these methods: bulk sequencing or single-cell RNA-seq or spatial RNA-seq. Another thing you have to consider before you start NGS study is the method: short read versus long read sequencing. Then, what is read length? NGS read length refers to the number of base pairs sequenced from the DNA fragments. The predominant difference between long read and the short read approach is the significant increase in read length. In contrast to short read sequencing, which comprises less than 300 base pair, long read has capacity to sequence on average over 10 kilobases in one single read, thereby requiring less read to cover the same gene. Computer programs are used to assemble the fragments into contiguous sequences. Read mapping is an essential part of the NGS analysis. Because the genome of most organisms is too long to be sequenced as one continuous string. Using NGS short-read sequencing, DNA is broken into short fragments that are amplified and then sequenced to produce reads. Usually, biomimetic techniques are used to piece together the reads like a jigsaw into continuous genomic sequences. So read mapping is the process to align the reads on a reference genome. This figure shows how to map the sequences. You will have a set of reads fragments, then you will align those reads into the reference genome. Long read sequencing has some advantages over short read sequences in mapping. Usually, The longer the read, the easier to map. You can see the difference between short reads and long reads. If you use the short read sequencing, there can be more chances of missing sequences data. That means there will be some sequence gaps. If you use the long read sequencing, long read map usually produces and covers larger variants and providing comprehensive variant detection. In case of some repeated sequences, short read sequencing can miss the number of repeated sequences. As you can see in this figure, there are 6 repeats. If you use short read sequencing, your result will tell there are just 2 reads. But if you use long read, you can tell there are 6 reads. There are many benefits of long reads, but it still has many limitations. There are many limitations of current long read technologies. Another issue you have to consider when you do the NGS study is the sequencing depth and coverage. Sequencing depth or read depth describes the number of times that a given nucleotide in the genome has been read in an experiment. Coverage describes a relation between sequence reads and references. This figure shows what is read depth and what is gene coverage very clearly. In conclusion, the higher depth it is, the better to use NGS analysis. Now we will go into the NGS study in urological diseases. NGS is accelerating urogenomics research by revealing the mechanisms behind complex urological diseases. The interplay between heritable and non-heritable mutations, epigenetics, and other factors require NGS-level analysis to increase our understanding. A wide variety of urogenetic diseases have been discovered owing to the development of NGS technology. NGS technology has led to great advances in understanding the causes of Mendelian or complex urological diseases. Owing to the complexity of genetic diseases, the genetic factors contributing to many rare and common urological diseases remain poorly understood. The clinical application of NGS significantly accelerated the discovery of disease-causing genes and promoted the understanding of molecular genetic mechanisms associated with hereditary diseases. Selecting the correct genetic test based on cost effectiveness, coverage area, and sequencing range can improve diagnosis, treatment, and prevention of urological disorders. Now I will explain about some of my experience in rare unidentified urological disease study. This study was initiated by enrollment of urological disease patients in an outpatient clinic, and basic medical evaluation was performed, and the pedigree study was performed. Whole genome sequencing using blood cell was performed, targeting the patients and their family. And when we found out the new mutations, the confirmation was done using Sanger sequencing. The first case was a 6-year-old boy. He come to the clinic with the symptom of dystonia and congenital blepharoptosis. At first, the diagnosis was ophthalmological disorders or urologic and neurodevelopmental disorders. But the clinician who take care of this boy suspected this disease will be some unknown urological diseases. So she interviewed with the family members and find out the mother has a mild symptom in the past, and the father was without symptom. During the whole genome sequencing, we identified likely pathogenic variant of SPAST gene. It was heterozygous, and this was observed in patient himself and his mother. We tried to do the further study, but because of the refusal of the patient, it was stopped. Another case was 23 years old man. He come to the clinic with palmar facial fibromatosis. The initial diagnosis was suspected rheumatological disorders or different diseases. His father has similar symptoms, and mother has no symptoms. When we performed the whole genome sequencing, we find out some new likely pathogenic variant of DDR2 gene, heterozygous, in the patient and his father. These cases, we also could not perform the further study. Another case was dementia cohort study. This study was done in a prospective cohort. The people over 55 years old from the community was enrolled. First, a medical evaluation was performed, and neuropsychological and cognition test was done for all the people who are enrolled, and whole genome sequencing using blood cell was performed. Blood and CSF biomarker was evaluated. These markers are the protein biomarkers, and image analysis using PET, MR, and functional MR was done. These two studies, the rare urological diseases and the dementia cohort study, was performed for more than 5 years, but it was stopped because the funding was terminated last year. During this NGS study, I can expect some future issues. First, NGS study is very cost-consuming study, and also we need big resources. So cost and resource matters is one of the biggest obstacle to perform the NGS study in urological diseases. These days, the wet lab cost is going down, and its speed is getting faster. So dry lab analysis becomes a bottleneck. In addition to the wet lab cost, so it is big issue to decide when do I start NGS study. Actually, during the study, I collected some patient samples, but some samples we didn't perform any NGS wet lab, and we still have many dry lab analysis to be performed. The second issue was integration of multi-omics data. At the beginning, we just planned the whole genome sequencing or whole exome sequencing, but we realized we needed to perform the transcriptome RNA-seq analysis and also epigenomic study are required in some cases, and we also have to add other omics like proteomics or metabolomics study. But we had some trouble in integrating these multi-omic data into a meaningful result. So we tried to develop the multi-omic integration and analysis pipeline, but it is just at the beginning stage now. Another issue was quality of the study. The quality of the sample for the NGS itself is very important issue. How to collect it and how to store the sample is very big issue. And in addition to the NGS data itself, we need to have plentiful of metadata. So how to access the metadata and the clinical data related with the patients or the normal people is another big issue. During the analysis and the wet lab, both in wet lab and the dry lab, standardization of the protocol and quality control of the data analysis pipeline was also big trouble. The other issues, what I realized that recently, is data sharing and disparity. We collected data for years, but we had a limitation in sharing those data because of some standard format and also accessibility, because our study was funded by the government and we don't have enough right to open those data. And also we had to get the permission from the patient or any people who provided the samples. And another issue was disparity, because the study which I performed is limited to Korean population. But when I want to compare the data with the other ethnicity, I recognized it has some trouble in direct comparison. And also, as you can see from many papers, the racial disparities or the basic disparities in designing the large cohort study has troubles. So I expect this data sharing and disparity will become a big trouble in the future. I briefly shared my experience in the NGS study in neurological disorders. Thank you for your attention. I really appreciate for me to have a good opportunity to present my experience in front of you. Thank you.
Thank you, Professor Dr. Hing Sohan, for such an amazing and informative talk. It was indeed a great presentation. Let us now proceed our next presentation. I would like to invite our third keynote speaker, Professor Dr. Tausen, Dean and Professor, School of Biomedical Sciences, Bachar University, China, and he will talk about deciphering molecules mechanisms of brain development and disorders in mice and humans. Please, Professor Dr. Tausen.
Hello, everyone. I'm Professor Tao Sang from Huazhou University in China. First, let me thank Professor Gulam Hossain for inviting me to give a seminar at this International Brain and Biomedicines Conference. I am so much honored to share our work with so many brilliant scientists at this conference. First, please let me briefly introduce myself. I did my PhD in neurobiology at University College London in United Kingdom. After finish my PhD, I did my postdoctoral training at Harvard Medical School. At Harvard, I worked with Chuck Stiles and Chris Walsh, those two superb scientists. In 2005, I was lucky to get a faculty position at Cornell University Biomedical College in New York, United States, and improved to associate professor. In 2017, I came back China and helped Huaqiao University to build up their first school or medical school and becoming the founding dean of the School of Medicine. Our lab has been working on the development of the central nervous system for many years, and in particularly the brain and the spinal cord. We are also very much interested in understanding the causes of human neurological diseases. Today, I want to share some of the work in human brain and spinal cord. And these diagrams shows the real brain images of human fetal brains at different stages. You can see gestational week 17 until 30. During this long process, you can see the morphology change and the growth of the brain, and the cell migrate, and the neurons form axonal connections. So brain is probably one of the most complex structures in human body. And we just look at the cell numbers, you can see there are over 100 billion neurons in the human brain. But no matter how complicated the human brain is, there can be two major cell types. One, we call them excitatory neurons. One, we call them inhibitory GABAergic interneurons. So if we look at the cerebral cortex, all the excitatory neurons, they are born from the region, we call it ventricular zone. They born from these progenitor cells. And how these progenitor cells proliferate and survive and differentiate is a major research interest to us. But if we look at the interneuron, they are also born from a structure called subpallium. And all those interneurons, they can be divided into three regions. For example, the median ganglionic eminence, MGE, in the subpallium, or lateral, LGE, and CGE. All those interneurons, once they are born from these three regions, they will undergo a long tangential migration and reach their final sites in the cerebral cortex. So recent years, people pay very much attention to the interneuron development because we are more and more realize that the balance between interneuron and excitatory neurons in the cerebral cortex is extremely important, because the unbalance between those two cell types could be a major reason of autism. This has been more and more recognized by scientists. If we look at the interneuron subtypes in the cerebral cortex, we can name them according to their morphology. For example, the basket cells, chandelier cells, they have beautiful morphology, they have many different cell types. But how these different cell types are specified in the human fetal brain, this is the question we are interested. In the past, it has been very difficult to address this question, but nowadays, based using the single-cell RNA sequencing approach, we are able to do it. Single-cell RNA sequencing, basically, you take a piece of brain tissues and dissociate those cells into single cells, and then you can use the 10x genomics platform and to sequence the RNA's transcription profiles in individual cells, and then gather information of one cell, and then you know the overall gene expression profile. To work on this project, two excellent students, Yuan Yu and Zhiwei Zheng, they worked on this project. What they did is that they collect the subpallium tissues from human and get their human fetal tissues from gestational week 9 to 12, and then use the single-cell RNA sequencing. And interestingly, you can say that those cells, they can be divided into several groups. That means there's a very high diversity of interneurons in human fetal brains. But no matter how complete they are, if we look at their gene expression profile and even look at their gene expression position using the in situ sequencing approach, you can see that they can be divided into two major regions. One is called the ventricular zone. You can see we use the pseudo color to label them in green. And one is in the subventricular zone. We use the purple color to label them. So this is very similar to the cerebral cortex. All those progenitor cells, they can be divided into ventricular zone and subventricular zone cells. And if we look at those interneurons derived from the MGE, you can see they can be grouped into 5 subtypes according to their combination of gene expression. And then those cells, they migrate tangentially into the cerebral cortex. So whether they are also specified, and when we look at their lineage development, we find one interesting feature is that they can be split into 2 lineages. We find that lineage 1 highly expressed gene like MEIS2, but if we look at the gene expression position of MEIS2, they mostly located in the dorsal region of the MGE. On the other hand, the lineage 2 highly expressed gene like LHX8 and ZIG4, they are located in the ventral region of the MGE. So this suggests that the interneuron precursor cells, they are positioned at different regions in the MGE, probably determine their migration route later on. If we look at the interneuron precursors in the LGE, they can be divided into 11 subtypes, but they also can be grouped into 2 types. One is in the dorsal LGE, one is in the ventral LGE. To the LGE specific interneurons, and if they are in the dorsal, they mostly are involved in the visual system development. And if they are in the ventral, they are involved in the neuron projection development. So the third question we look at is that the diversity of interneuron precursors, how they are specified at later stage or early stage. On the left is the diagram of different cell types in the cerebral cortex. You can see they are located in the layer 1, layer 2, 3, and layer 5. But when we collect their gene expression profile with their adult marker, we find there's a very nice connection even at gestational 9. So this suggests that the diversity of interneuron neurons, they have a very early acquisition. That means their cell fate or cell types are predetermined even at fetal stage. So we published this work by end of the 2021 in Nature Neuroscience, and we have drawn 3 major conclusions. The first is that interneuron progenitors and precursors in human fetal subpallium are very high diversed. And the second is that we find the interneuron precursors in the MGE and LGE, they simultaneously acquire their temporal and spatial identity. They can be divided into dorsal and ventral subtypes. And the interneuron subtypes can be specified even at fetal stage. So this suggests that there's an early acquisition of cell fate for the various interneurons. So in the end, I want to share some unpublished work in the human spinal cord. And you can see human spinal cord, there's two major cell types. One is motor neuron and one is interneuron. So the human spinal cord neurons are very complicated. They have about 20 distinct cell types. So the question to us is also how they are specified. So a PhD student, Jing Wang, worked on this project. She collected human fetal spinal cord from gestational week 8 to 12 and used the RNA sequencing approach to analyze their cell diversity. And you can see again, it's very similar to the subpallium. They can be divided, into different cell types at very early stages. So, for example, they're just interneuron and motor neuron. They can be divided into 13 subtypes, and they have the progenitor for motor neurons and also progenitors for interneurons. But when we analyze those progenitors very carefully, we find one interesting group of cells. We call them common progenitors. They can be specified in 5 subtypes. Interestingly, we find that these common progenitors, they can give rise to motor neuron progenitor type 1, and then motor neuron progenitor type 1 will become type 2 and type 3, and later on mature into motor neurons. And these common progenitors also can give rise to interneuron progenitors, and then these interneuron progenitors will differentiate into various interneurons. So interneurons are very complicated in the human spinal cord. There are at least 11 interneuron subtypes aligned from the dorsal to the ventral spinal cord. So what about their gene expression features? Interestingly, when we look at interneurons, we find they can be grouped into 13 subtypes. And particularly, when you look at all those gene expression profiles, they correspond very well with their position in the spinal cord from the dorsal interneurons and to the ventral interneurons. So this suggests that even though the interneurons are very complicated in the human fetal spinal cord, they are already specified by combination of gene expression at very early stages. So to summarize my talk today, and we find that there are 2 types of neuroprogenitors. They reside in the ventricular zone and the subventricular zone. They are very similar in the cortex, subpallium, and spinal cord. So probably this is the common theme of central nervous system development in the human tissue. Second is that we find single-cell RNA sequencing can be a powerful tool to capture the diversity of neuronal subtypes in the human fetal brain and spinal cord. And third, we find that the cell fate determination can be a very early event. So it happened very early in the human fetal brain and predetermine their fate for the adult stage. So I'd like to thank all the people in my lab, and we have 2 lecturers, and the rest, they are all PhD students and master degree students. They work very hard to generate all those results I've presented today. And I also want to thank the funding support while I worked in the United States. For example, the National Institute of Health, and also when I come back to China, the National Natural Science Foundation of China. And in particularly, I want to thank all my collaborators, Greg Hannon at Cold Spring Harbor, Tyler Jacks at MIT, Yunan Jian at UCSF, while I worked in America. And after I came back China, I was so fortunate to set up a very active collaboration with Professor Ghulam Hossain at Government College University in Pakistan, and Wenjie Cai from Fujian Medical University, and Zhi Hengxu from Chinese Academy of Sciences. And thank you again for attending my seminar. I'm so honored to share my work with everybody here. Thank you.
Thank you, Professor Dr. Towson, for such a remarkable talk. Now, the plenary session has ended, and I am thankful to all the keynote speakers for their valuable insights. Now we will have our first oral presentation session, which will include talks from the field of anatomy, physiology, and epidemiology. Thank you for your participation. Stay tuned with us. Thank you.